It's a real pleasure to have Marlene here. Marlene is a professor at the University of Pittsburgh and associate director of the Center for the Neural Basis of Cognition. She did a PhD at Stanford with Bill Newsom and then did a postdoc with John Monsell at Harvard, both big names. And over the last 10 to 15 years, she has established herself and her lab as one of the undisputed leaders in recording from populations of neurons and rigorously linking their activity to very well characterized behavior in non-human primates who are doing difficult cognitive tasks. So I'm really looking forward to her talk. She's won numerous awards, including the Klingenstein Fellowship Award, the Eppendorf Prize, McKnight Scholar Award, and the Trollin Research Award from the National Academy of Sciences. So over to you, Marlene, and we are all eagerly looking forward.
Great. Thank you so much for that way too kind introduction, and especially for the invitation to talk to all of you. I'm going to talk today about some brand new unpublished work that I have never talked about in public. And my lab has been going in some new quantitative directions. And so I'm really excited for the opportunity to talk to all of you about this. Because this is so new, and in general, I would be really grateful for your feedback and questions. So as we're going, if you would like to write questions in the chat or interrupt me if something's unclear, I'm happy to take that anytime. All right, so the big question that my lab studies is the same one that many of the people I see in the audience study, which is how is information about our world, in our case, visual information, encoded in the brain and used to guide behaviors. And the reason that we don't know the answer to this question, despite many people working very hard on it, is that it's pretty complicated. Many aspects of it are complicated. The first is that behavior, in particular the behavior of primates, is really complex and amazingly flexible. So you can respond to many different visual aspects of your environment. You can do so in a flexible way that depends on the context that you're in. And you can juggle many different tasks at the same time. And so one of the most important aspects of this system is that it's not static, and it depends on many aspects of our cognitive states, which are both really interesting and exciting to study, but also challenging to study. And then maybe the bigger part of why this is a hard problem is that flexible, complex behavior is mediated by a really complex structure. And the complexity of the human brain is rivaled by only very, very few systems in our universe. And there have been a number of interesting recent opinion pieces comparing the complexity of the human brain to that of the structure of the observable universe. So even though, of course, there are something like 29 orders of magnitude separate this thing from this one in size, the complexity is similar when measured in particular ways. And one way of quantifying that in a really crude way is just that the universe is thought to have about 100 billion galaxies, which is about the same as the number of neurons, which are the unit of computation in the human brain. So this is a complicated thing, and we're studying how it does complicated things. And so our basic strategy for addressing this problem is to record from the brains of monkeys who are making visually guided decisions. So we think this is a good strategy for a few reasons. The way this works is that we bring rhesus monkeys into the lab. They sit in front of a computer monitor. We show them visual stimuli, often fairly simple ones that we can control easily. And we teach them to do tasks in which they tell us something about what they see. And while they're doing that, we record the activity of a few dozen neurons in their brains. And while this is, of course, a recording from a very, very small subset of those 100 billion neurons, we do this because knowing something about how active, and I'll quantify that as the number of action potentials or spikes that each neuron fires at each given moment. And by knowing something about those patterns of activity, we can get a snapshot of the neural computations that are going on at any given moment, and that allow the monkey to perform these tasks. And the reason we think this is a fairly good strategy is that it has allowed us and many others to form links between the activity of groups of neurons in the brain and many other ways that the brain is usually studied. And I've drawn these as islands in some ocean of neuroscience because often they are studied separately. So our lab has looked in particular at links between the activity of populations of neurons and aspects of the monkey's cognition through our collaborations with our colleagues. We have also used them to constrain mechanistic models of the underlying mechanisms that might cause changes in the brain when things like cognition change. We use computational methods to assess the neural computations performed by these groups of neurons. And then, of course, we measure the monkey's behavior. And overall, I would say that this vein of study has been successful in that we can find links between neural activity and each of these. The problem is that when we start to study complex behaviors and more of the neurons that are involved in those behaviors, those links become pretty complicated, and our simple quantitative ways of quantifying the activity of groups of neurons might allow us to link the brain to each of these individually, but we are lacking a comprehensive framework that would help us to understand the links between all of them as a whole. And so what I'm going to talk to you about today is some of my lab's new efforts to do that. And maybe the punchline of the talk is that the quantitative framework we've been using, which is called topological data analysis. I usually hate using acronyms, but I'm going to say TDA because if I have to say topological data analysis too many times, this talk will take forever. So we think that TDA, which is a mathematical method that has been used for several applications, but maybe most notably astrophysics, might turn out to be the bridge between these different levels of studying the brain. So the idea is that we're borrowing methods that have been used to help to understand the structure of this complicated thing. And we're going to see what we can learn about the structure of this other complicated thing. Okay. So what I'm going to do for the rest of the talk is I'm going to start out by telling you about some of our most relevant results in these different ways of studying the brain. I will then point out why our existing quantitative frameworks are insufficient, and then start to tell you about how TDA can in fact be a bridge between all of these different levels of study. Okay, so let's start by talking about cognition, because as I mentioned, perhaps the most remarkable and also important aspect of visually guided behavior is how flexible it is. So we know that there are many aspects of cognition that can change everything about the way you see and perceive a visual scene. And in particular, if you are trying to do some sort of visually guided task, you can improve your performance on that task using a number of different cognitive processes. So these are illustrations of 4 of the cognitive processes that my lab has studied. You can see things better by selectively paying attention to a particular part of a visual scene. You can do better through learning or practice. You can change your performance using very general cognitive processes like alertness or arousal. And of course, you can change what you do with that information by switching tasks. We know that all of these processes not only affect performance on the kind of tasks that we train our monkeys to do, they also affect the activity of neurons throughout the brain, and in particular in visual cortex. So this is an example of the type of task we use in the lab and how we might manipulate one of those cognitive processes with monkeys. In this drawing, each of these rectangles is an illustration of what is on the computer monitor that the monkey is sitting in front of. And a trial begins when the monkey fixates this central spot of light. And then 2 of these oriented stripy stimuli called the gabors start flashing on and off. And at a random and unsignaled time in the trial, the orientation of one of those 2 changes slightly. And the monkey's job is to tell us that he noticed that change by looking at the stimulus that changed within a few hundred milliseconds. And we can manipulate attention, or rather spatial attention, that is which part of space, which part of the visual world he's paying attention to, by cueing the monkey in blocks of trials as to which of these two stimuli is most likely to change. And so if it's an attend left block of trials like I've drawn here, the monkey has learned that what that means is that on 80% of those trials, the stimulus on the left is going to be the one that has changed. But the monkey is rewarded for detecting a change at either location. So it turns out that manipulations like this have a really big effect on the monkey's behavior. This is an example psychometric curve or plot of the monkey's proportion correct as a function of how easy or how difficult the change was to detect. And this is just one day of data, but it is really representative of performance in these sort of tasks. So what you can see is that when that stimulus change is very big, it's obvious, and the monkey almost always detects it correctly. When that stimulus change is really subtle, like only 1 or 2 degrees, he rarely detects it, and then everything in between. These green points are the trials where the change happened in the cued location, meaning that we told him to expect a change on the left, and in fact, that's where it happened. This blue point is when we fooled the monkey. That's the 20% of trials where we told him to expect a change on the right, but it actually happened on the left. And you can see that for this example session, which again is pretty representative, that attention improved the monkey's ability to detect something like 10-degree orientation or also motion direction change from something like 40% correct to nearly perfect performance. This is a really big deal. And especially if you're a monkey who is sitting there playing this game, because when you successfully detect these things, you get a fruit juice reward. This improvement from 40% to nearly perfect performance is a big deal. It's a lot of fruit juice. And what we're trying to do is understand what are the changes in the brain that might cause this big change in behavior. Now, maybe a summary of the past decade or so of work in my lab and others is that there is one measure of neural activity that's been pretty good at accounting for that improvement in behavior. What we found by studying all 4 of these is that when in cognitive conditions where the monkey's performance changes, so does what I'm going to call correlated variability in visual cortex. So there are a number of ways of measuring correlated variability. This is the simple one that we use. So if we imagine that we are recording from 2 neurons in visual cortex at the same time, what we know about neurons in general is that their responses are variable. So we could show the monkey the exact same visual image on many trials, and we know that each neuron will fire a slightly different amount each time the monkey sees that identical stimulus. And so I'm going to use a measure called noise correlation, which is also called spike count correlation, sometimes abbreviated as RSC. Those words in literature all mean the same thing. And that's a way of quantifying the extent to which those trial-to-trial fluctuations in response to the same visual image are correlated between a pair of neurons. So we might show the monkey the same image a bunch of times, and we record the response of neuron 1 on every trial. We'll plot that on the x-axis, and neuron 2 on the same trials on the y-axis. And in this hypothetical example, the trial-to-trial fluctuations in their responses are highly correlated, meaning a trial where neuron 1 happens to fire more than its average, neuron 2 is doing the same thing. But you could imagine doing the exact same experiment and finding that the trial-to-trial fluctuations in their responses are completely independent. And we quantify the relationship between those fluctuations by simply taking a correlation coefficient. In visual cortex, if you record from 2 neurons that are sort of near each other in the same brain area that encode more or less the same things, what we typically find is that the noise correlations are positive, although not nearly as positive as I've drawn here. And what we find is that in conditions when the monkey is doing better, like when he's paying attention to the part of visual space that those neurons both encode, the responses become more independent. So we go from a situation where the responses are positively correlated to closer to independent. Amy Nee, who's a terrific postdoc in my lab, quantified this effect. And in a study where she varied both selective or spatial attention and perceptual learning. And she found that there was a strong quantitative link between the average correlated variability in visual cortex and the monkey's performance on her task. So what she did was she recorded from groups of neurons in visual area V4, that is a mid-level visual area where we think the neurons are responsible, at least in part, for your ability to do tasks like this. And she recorded both while she cued the monkey to attend within and outside the receptive field of the recorded neurons, meaning attending to the thing they both care about versus not. And she also recorded from the same neurons throughout several weeks or months as the monkey learned to become better and better at detecting more and more subtle changes in the visual stimulus. So she could take the same data, divide it different ways, and see the effect of attention and see the effect of learning over long periods of time. And what she found is that the average noise correlation between all the pairs of neurons she recorded simultaneously was strongly related to the monkey's ability to do this task. So these are two plots of the exact same data colored differently. On the y-axis is the average noise correlation. So again, she's recording from a few dozen neurons in visual cortex. She just calculates the noise correlation between each pair of neurons and then averages it. And then on the x-axis is a measure of the monkey's behavior. This is sensitivity in a signal detection sense for the aficionados. So big numbers mean good performance. And just like other studies had shown, she found that attention improves performance. So you can see the black points here are generally to the right of the gray points. And learning also improves performance. The blue points, which are late in learning, are generally to the right of the red points. And she also found that both of those affect noise correlation. So again, the gray points, unattended or uncued, are mostly above the black points, and early in learning, red is mostly above the blue points. But what hadn't been seen before is that this wasn't just a general trend, attention and learning both improve performance and reduce noise correlations. Instead, there seems to be a single relationship between the magnitude of noise correlations and the monkey's performance. So these best fit lines through different splits of the data are statistically indistinguishable. So what that means is that for this monkey, on average, when the noise correlation was, 0.2, the monkey was doing the task at a D prime of about 1. And that was regardless of whether he got there because, say, he was attending but early in learning or not attending but late in learning. Okay, so a single relationship between this measure of neural activity and behavior. So this was pretty great. It explains actually a surprising number of things. Amy's been doing some new experiments, giving monkeys drugs that are used to treat disorders of attention, like the active ingredient in Ritalin. And just like this, she finds that those drugs affect behavior exactly when they affect noise correlations in V4, according to these same lines. So that's pretty good for a very crude measure of neural activity. But probably it won't surprise you to learn that this really crude measure, average noise correlation, starts to fall apart in a few more complicated situations. I'm not going to go into the details today, but we've also been experimenting with more complex visual stimuli, those that are more naturalistic, and richer behaviors. This is an example of a task where the monkey is asked to tell us about motion direction by making an eye movement to a ring, so we get a continuous measure of behavior. And in these situations, the average noise correlation, the mean correlated variability, doesn't do a very good job of explaining the details of behavior, especially when confronted with rich naturalistic visual stimuli. This simple measure also doesn't allow us to precisely identify the biological mechanisms that are responsible for that improvement in performance. So our collaborator, Chung Huang, who's in the Department of Neuroscience at the University of Pittsburgh, has created what we think are really terrific biophysically inspired models of visual cortex. So I very much encourage you to read her papers, but the guts of this is that she models populations of neurons that respond to visual stimuli that are in different brain areas throughout the visual pathway. And there are both excitatory neurons, where increases in their activity increase the activity of their surrounding neurons, and inhibitory neurons, where increases in their activity inhibit the activity, reduce the activity of surrounding neurons. And so what she did was try to figure out, in the context of her model, what are the changes in this circuit that could cause the changes in noise correlations that we see. And what she found is that it turns out there are two mechanisms that do just about as well. So one thing I haven't told you that yet is that in addition to reducing noise correlations, cognitive processes like attention and learning change the dimensionality of noise correlations. And so Chung plots this here for Amy's data. Sorry, these are for outputs of her model, of Chung's model. So what she is plotting here is the shared variance on the y-axis, and this is normalized by the amount of variance in the first dimension, or the first eigenmode. And she's plotting that shared dimension, that shared variance for each of the first 5 eigenmodes. And so in this sort of plot, this a more flat line shows you that the structure of noise correlation, correlated variability, I apologize, is more high dimensional. If this is a more steep drop off, as you see here, then that tells you that most of the variance is in that first eigenmode. In her model, she can change the average activity of the inhibitory neurons, and that both changes the mean noise correlation, and it also changes the dimensionality of the variability in ways that are qualitatively and quantitatively similar to the data that Amy recorded. But it turns out she can change something else about her model and get basically the same answer. Here, she's changing the time scale of inhibition, the time scale at which these neurons functionally affect the neurons around them. And reducing that time scale causes changes in correlated variability that are, again, very similar to what we see with attention, and very similar to changing the mean activity of those inhibitory neurons. And so there are two mechanisms here which both change correlated variability in ways that are similar to the data, and in ways that are indistinguishable using traditional linear measures of correlated variability. So for this and several other reasons, we feel that we need a richer quantitative way of describing the structure of that shared activity of large populations of neurons. And so that is where topological data analysis comes in. And everything else I'm going to talk about today is the work of a really phenomenal graduate student in my lab named Tevin Rouse. Tevin has bachelor's and master's degrees in mathematics. He has a long-standing interest in the field of algebraic topology, which is where these analysis methods came from. And as I mentioned earlier, mostly these methods have been used to describe very different kinds of systems, for example, the structure of the universe in astrophysics. Tevin realized that there might be, that we might learn something qualitatively different using these methods to quantify the structure of noise correlations in our data. So these methods are profoundly nonlinear, and they are really orthogonal to the sorts of linear methods that we and others had used previously. So what Tevin does is he starts with this very same noise correlation matrix that I've been describing. This is a number of neurons by number of neurons matrix. So the number of neurons is the number of neurons that Amy recorded simultaneously. And then the color here indicates the magnitude of that noise correlation. So dark colors are more highly correlated pairs. And of course, what I've shown you so far, Amy showed that the behavior is related just to the average across all these pairs. Chung showed that attention changes the dimensionality in as assessed by factor analysis using linear methods. Tevin assesses this matrix in a very different way. And so I'm just going to show you a really simple illustration to try to give some intuition for these methods. So the first thing he does is plot these data in a space in which each point is one neuron, and the distance between any two neurons is 1 minus their noise correlation. So of course, so in this metric, if two neurons are very highly correlated, they will be very close together in this space. If they are uncorrelated or even anti-correlated, then they'll be very far apart. And for populations of any reasonable size, in order to make a plot like this, that space needs to be higher dimensional. I am just showing you a two-dimensional illustration right now. So this is an illustration of maybe 7 neurons, and the distance between them tells you about 1 minus their noise correlation. And then we could, the guts of this analysis is to simply iterate through different threshold values of this distance metric and declare that two neurons are functionally connected if their distance is less than the threshold value. So for very small values in this illustration, none of my neurons are connected because none are very, very close together. But if we get to a threshold that's high enough, we might see that neurons are connected to their nearest neighbor. We could increase the threshold a little bit more and include this neuron. And now these 3 neurons are all-to-all connected. And so we sort of color that in to signify that there are no holes in this subpopulation of neurons. And then of course, as the threshold increases, eventually by necessity, because this is a bounded space, at a high enough threshold, every neuron will be functionally connected to everyone else. And what we can do for each level of that distance threshold is simply count the number of holes. So at very low thresholds, no one was connected to anyone else, so there are no holes. At very high thresholds, every neuron will be connected to every other, so no holes. But at these intermediate values, we have 1 hole in this toy example. I see a question in the chat. Is this one? Yes. How do you decide what threshold to use? We iterate through all of them. So since this distance metric is bounded, since the lowest the correlation value can get is -1, so it's bounded at 0 on one end and 2 on the other, we just iterate through every threshold value. And we can summarize the results of this analysis using a plot like this. This is called a barcode in TDA speak. So we can iterate through all values of the distance threshold. This is normalized 0 to 1. And again, for this toy example, at very low values of the threshold, we have no holes. At intermediate values, we have 1 hole. And then at high enough values, we have no holes again. And so we have just 1 line indicating that we have this 1 hole that persists for some range of threshold values. The traditional uses of TDA, including to study the structure of the universe, focus on what are called persistent features like this. We can learn something about the arrangement of these neurons in this toy example, that at least some of them are kind of arranged in a circle, by looking for individual holes that persist over many values of this threshold. In the real neural data, though, that is not what we see. So this is a barcode for 1 data set in 1 attention condition. Now, each of these horizontal lines indicates 1 hole, and you can see as a function of the distance threshold. And what you see is that there's no 1 hole that's very remarkable. We don't see something that lasts across a very broad range of thresholds. Instead, we see many holes, and they begin and end at a range of threshold values. And this turns out to be very characteristic of all of the data all of the neural data that Tevin has looked at. It is very different than any other use of TDA that we know of. So these are structures that are pretty different. This is not telling us that all of our neurons are arranged in a ring. Instead, it's telling us that it's a complicated structure with a bunch of holes that don't last beyond a very limited range of threshold values. So in my illustration, I told you about a hole that is topologically equivalent to a circle, meaning that it is a sort of a circular arrangement of these neurons, even though that circle might be bent or stretched in different ways. And that's what I've illustrated here. For the topology aficionados, we would say that those are the holes detected by homology group 1, H1 here. We could also look for holes that are topologically equivalent to a sphere, or spheres in higher dimensions. And we could also look for holes that are really not holes at all. They're topologically equivalent to a point. And those are denoted by these different homology groups. Another difference between our neural data and many of the other applications of TDA is that we don't see features that are particularly unique to one homology group. In fact, this pattern where we see a bunch of holes that don't persist very long, that's true in all of the homology groups that we have enough data to measure. And so I'm going to show you the different homology groups in just a second. And then mostly focus on just the one for which we have sort of the best measurements. But everything we learned about, for example, the relationship to behavior or attention is true in all of the homology groups that we were able to measure. Okay, so we could summarize this sort of barcode by just kind of taking an integral under it, counting the number of holes at each threshold level. And that is called a Betti curve, after the Italian mathematician who pioneered some of these methods. So these are Betti curves for some of Amy's data in the attended and unattended condition. So again, we're plotting the number of holes on the y-axis as a function of that distance threshold normalized. The different curves here are for the different homology groups. And then the different plots are the two different attention conditions. And what you can see for this example session is true across the data, that we see these distributions of Betti numbers, of number of holes across fairly restricted ranges of distance thresholds, except for homology group 0, which is just lots of points, individual points that last for a long time. So we see these distributions, and we see that attention changes those distributions. In particular, in the attended condition, there are more holes. They are restricted to a tighter range of distance thresholds. And therefore, the peak of these distributions tends to be higher. And so I'm going to summarize these distributions for each recording session by just taking the peak of these curves. And we have tried a number of other common topological descriptions of the data. They all do more or less the same thing in terms of their relationship to the other measures that we've looked at. So I'm going to focus for today on the peaks of these Betti curves. Okay. So now let me tell you about how these topological measures can form a bridge between all of these different ways of studying the brain. I showed you already for that one recording session that there is a link between the peak of the Betti curve and the attention state. Let me show you that for all of the data now. So what I'm plotting here is this measure that we were talking about, the average noise correlation. And what is on the y-axis is the peak of that Betti curve. And I should say that these values have all been z-scored because that allows us to combine the data across our two different monkey subjects. So the two different symbols here, the points and the triangles, are the two different monkeys. And then the two colors are the attended and unattended conditions. So every recording session contributes 2 points to a plot like this, one in each attention condition. And you can see that there is a strong relationship between the average peak of the Betti curve and the average noise correlation. You can also see that attention changes both those numbers. What you can see is that in general, the red points are above and to the left of the yellow ones. So the peak Betti number is related to mean noise correlation. It is therefore unsurprising that it is also related to attention. And we also see that just like noise correlation, although the function's a little bit different, there's a relationship between the peak of the Betti curve and the monkey's behavioral performance. Again, using this same D prime measure, but now z-scored so that we can combine across monkeys. And again, you see this positive correlation between these, meaning that when the monkey is doing well, whether that's because of the attention condition or just because he happens to be doing well that day, the peak of the Betti curve tends to be higher. So we see that this topological description of the neural data is related both to attention and to overall performance on this task. We have also found that the peak of the Betti curve is a much better way than these linear descriptions at distinguishing between the two biological mechanisms in Chung's model. So just to remind you, we have this circuit model of neurons in different visual areas. We have inhibitory and excitatory neurons. And Chung was able to mimic the effects of attention on correlated variability, at least as assessed using linear methods, by either changing the average activity of the inhibitory neurons or the time scale of inhibition. But it turns out that these two different mechanisms make very different predictions for the pattern of changes in the peak Betti number. So that's what I am plotting here. Should put that plot just right in the place I can't see it because of the Zoom window. Sorry about that. So on the x-axis here is the normalized shared variance, and this is the eigenvalue for the second mode divided by the first. So that's kind of these plots here. And the y-axis is the peak Betti number. This purple curve describes the results in this left-hand plot. This is changing the mean of the inhibition. And just like we see in the data, this proposed mechanism for attention increases the peak Betti number. When Chung tried to change or did change the magnitude of noise correlations using the time scale of inhibition, she had to work really hard. So these first few points here are all of the points on this graph. So anything in a physiological range in terms of average noise correlation had no effect on the peak Betti number. And when she pushed the system into an asynchronous state, which is really unlike what we see in physiology, she did see a very rapid increase in the peak Betti number. And so this slower change in both the shared variance and in the peak Betti number is much more like what we see in the real data. And we are excited about some future work in collaboration with Caroline Runyon, who is someone who uses optogenetics in mice, where we are in fact mimicking this mechanism, changing the activity of inhibitory neurons. And in fact, in our preliminary recordings, we do see these increases in the peak Betti number. Says lost track of the peak Betti number, how it's calculated. Yes. So let me, oh no. So the peak Betti number is just the number of holes at peak, the peak number of holes at any distance threshold. So here are our what's called Betti curves. It's just a plot of number of holes as a function of that distance threshold. And again, the distance metric is 1 minus the pairwise, the noise correlation between two neurons. And so again, we get holes like this. We iterate through this distance threshold. We figure out the range of thresholds over which each hole persists. And then we just simply count the number of holes at each threshold to generate these curves. And the peak Betti number is just the peak of that curve. This precise measure really doesn't matter all that much. We can use many other measures of this, for example, the total number of holes, and then some other more sophisticated measures of the topology of these spaces and get more or less the same answer. All right. Okay, so we've now seen that the topological description of this data is pretty good for differentiating between attention conditions. It helped us distinguish between potential biological mechanisms in ways that our more traditional linear methods did not. And then we wanted to see if it could also help us understand what these networks of neurons are doing. What is the computation performed by these networks? And in particular, how does that change with attention? So to do that, Tevin brought in some ideas from network control theory. And this is a whole area of work that has been used to assess the functions of all sorts of networks, mainly electrical networks. There has been some really beautiful work by Danny Bassett taking this into the realm of neuroscience. She's mostly focused on much bigger networks than I'm talking about. For example, using functional MRI in humans, assessing the relationships between whole different areas of the brain. Her work is really, really terrific. And I saw that she is your speaker for this seminar series next week. So I definitely encourage you to check that out. She really is pioneering in this area. We use this idea with Danny's great help in a slightly different way. Again, to look at this matrix of pairwise noise correlations between the groups of neurons, between the neurons that Amy recorded in visual cortex. And so the guts of this is that to calculate what's called an energy landscape, which plots the amount of energy it would require to take the network between any two states. And this is constrained by this correlation matrix. So as a thought experiment, just imagine that all of our neurons are very highly correlated. That means the whole population would be moving up and down together. That means that if you took any old starting state, for example, the response to one of the Gabor stimuli in our task, it would be much easier to get to some other states than others. So if we had some starting state, if all the neurons are moving up and down together, it would be pretty easy to get to a state where all of the neurons increased or all of the neurons decreased their rate. On the other hand, some states would be very difficult to get to, because if all the neurons are moving up and down together, it would be hard to get to a state where, say, half the neurons are firing a lot and half are firing very little. Okay? So this is a way of calculating across all possible states of the network how easy, dark colors in this silly illustration, or how difficult, lighter, more yellowy colors here, it would be to get to any other state. And there are two measures of the overall energy landscape that Danny and others have used to quantify this overall landscape, the overall state of the network. One is called average controllability, and that quantifies how much energy it takes to move from one state to a nearby state. And so in our case, we might think of that as the amount of energy, or how easy or difficult it is to move from, say, the response to the starting orientation in our task to a subtly changed stimulus, maybe one that's also one of the changed stimuli in our task. There is a separate measure called modal controllability, which measures how easy or difficult it is to move the network from one state to a very different state. And we might think about this as maybe moving from a state where the monkey is focused on his stripy Gabor stimulus to one where he's thinking about something different altogether, like bananas. Okay, so these are two different measures. High average controllability means it's easy to move between nearby states. High modal controllability means it's easy to move between very different states. And mathematically, and also in our data, these two measures are independent, meaning they can both be high or they can both be low, or one of each. Okay. So first, what Tevin computed both analytically and then saw in the data is that both average and modal controllability are related to these topological descriptions. And so I'm showing you here a plot of average controllability, again, how easy or difficult it is to move between nearby states, that's on the y-axis, as a function of that peak Betti number. And again, you see a strong relationship, and in fact, controllability is changed by attention. We also saw a relationship, although a stronger one with other topological measures that I'm not showing you, between modal controllability and topology. So here, again, we're plotting the modal controllability. High numbers mean easy to move between very distant states as a function of that peak Betti value. And you can see very easily that attention has a strong effect on modal controllability. So we are excited, we're just scratching the surface of this. We're excited about the potential for these measures to help us understand the function of the network. I can show you one early return, which will then tie into behavior, which is that we see that both average and modal controllability are associated with behavior, with the monkey's ability to do our task, but with different aspects of behavior. So if you recall, average controllability is how easy it is for the network to move between two nearby states. For example, responses to two of the different stimuli that the monkey needs to tell the difference between. And so what we did was we took all the trials in this data set, we sorted them by whether the network was in a high and blue or low controllability state. And then we divided it into, I don't know, maybe 7 bins by controllability. So high controllability in blue, progression to low controllability in red. High means easy to move between these two states. Low means hard. And what you can see is that this was related to the monkey's performance in our task. So here is the monkey's proportion correct as a function of how big or small the orientation change was. Easy changes on the right here, very subtle, difficult changes on the left here. Of course, the monkey's performance is always better for easy than difficult trials. But what you can see is that basically across the board, the monkey is doing better when the network happens to be in a high controllability state, as defined by the status of the noise correlations. Yep. So the question is, can I please repeat how the controllability measure was computed? What it is for every possible state of the network, and a network state is defined by the vector of firing rates of all of the simultaneously recorded neurons. The question is basically, what is the probability? It's related to the probability of moving from one starting state to any other state. And average controllability is related to the ease of moving between nearby states. Modal controllability is related to the ease of moving between very distant states. And this is constrained by the noise correlations, because in a situation, because that tells you how the neurons change together. So for example, in a situation like the unattended state, where they're all moving up and down together, at least to a certain extent, that means it's very easy to move between states where, for example, maybe your starting state is one where all of the neurons are firing at a medium amount, and maybe they could all increase their rate or all decrease their rate. But when they are highly correlated, it's more difficult to move to a state where, for example, half increase and half decrease. There are formal definitions in Danny's paper, and I am happy to pull those up at the end if that's okay. Okay, so what we see is that average controllability, which is the ease of moving between two nearby states, is associated with behavior basically across the board at all orientation change amounts. We've got a floor effect over here, but you see this more or less ordering from blue to red in all cases. And then we went to look at modal controllability. So again, modal controllability is something different. That is the amount of energy or the ease of moving between one state and a very distant state. And what we see is that modal controllability was only associated with differences in behavior for the very easiest trials. And it's sort of backwards, meaning more controllable, that's blue, was worse performance on these very easy trials. And our hand wavy explanation that we are very excited to explore in tasks that are give us a little bit more rich behavioral information. Our hand wavy explanation is when your brain is in a state where it's really easy to move between distant states, like then maybe you're more prone to things like mind wandering. So the monkey maybe is sometimes should be focusing on his gabors, but he's thinking about the banana he's going to eat when he gets back to his cage. And that may cause lapses in what would otherwise be very easy tasks. And so in psychology, the imperfect behavior, even on easy trials, is called a lapse rate. And what we find is that this lapse rate is related to modal controllability, meaning more controllability, easier to move between states, maybe more mind wandering, is associated with worse behavior on these easy trials and really no difference, maybe even a slight reordering on the hard trials. Okay. All right. So overall, what I've told you about are what we view as really the beginnings of this work, this effort to find a quantitative framework that would allow us to link in a singular way many different ways that people study the brain. I told you a little bit about the link between topology and cognition as measured by selective attention. I showed you that unlike our earlier easy linear methods, the topological descriptions of correlated variability could help us distinguish between mechanisms in our models. They tell us something about the function of the network and about why these different states might lead to changes in behavior. So as I said, we view this as very much a starting point, but one that we are pretty excited about. And so with that, I would like to thank the members of my lab who contributed to this work. Amy Mee did the experimental work. Tevin Rouse did the topology and most of the analysis work. If you find these questions or approaches interesting, my lab is planning to move to the University of Chicago starting this summer, and we will be hiring at all levels there. So if this sounds interesting, please get in touch. And many thanks to all of you for your attention.
Thanks, Marlene. That was a fantastic talk, of course, as expected. Eric has a question about TDA.
Yeah, hi, Marlene. Very good. I love your work and all the stuff you guys are doing. And this problem of, how do you understand population activity and how do you put a framework around it? And that's very interesting. I didn't quite understand it. I was going to ask you a question about just the very basics of your TDA. Have you guys explored what the sort of the default random model would give you? In other words, if I just randomly assign spike count correlations between two populations where spike count correlations are a little higher for the unattended and a little lower for the attended, do these analyses, what do they tell you in the random sort of control?
Yeah. So what you get, so we did almost that exact thing. So we and others have seen that the correlation structure is amazingly low rank or low dimensional. And so we experimented with just taking, fake neurons and imposing one-dimensional correlation structures or iterating up like that. And in those models, you get just tons of holes, like orders of magnitude more than we see in the real data. So
really,
while it is true that attention changes the number of holes and all those things, and I wanted the title of our paper to be "Attention is Holy," but that got nixed very quickly. It's actually true that our toy models give just zillions of holes. And one thing that's kind of interesting is that both in the real data and Chung's biophysically inspired model gives sort of realistic numbers of holes. And whereas more simple models that are not as constrained by the biology do not. So we don't deeply understand that, but I think that one thing is that the neural data have many fewer holes than you would expect just from a toy model.
Well, that's very interesting. That's great.
So question from Paul.
Yeah, so I had a question. I also really found this fascinating and very intriguing. I can't claim that I actually understood everything, but one question that I have though for you is that When you're recording the neurons, of course, you're really highly subsampling the real population. And so the question, of course, is then how sensitive are the conclusions you get from this sort of analysis to having gotten the right neurons and enough of them? And can you make some kind of a scaling argument? How confident can you be in the topological conclusions on that basis? And also, for example, of course, the possibility of certain types of neurons just being really underrepresented simply because they're small and hard to record, et cetera, et cetera.
Yes. So as you might expect, my answer to your question is going to be a little bit unsatisfying, but we're interested in that question and we have been moving toward it. So here are a few things we've done. One way is in the models. There you can obviously model a much larger number, and so we can ask, how do our conclusions about the topology depend on like the proportion of modeled neurons that we subsample, and in the range that we can assess, and then also by subsampling our own data, we think that the absolute number of holes that you measure absolutely depends on the number of neurons and firing rates and average correlation and things like this. But the general trends that we see do not. So that has held up pretty well, with the caveat that these topological computations are really computationally expensive. So Tevin's got the Pittsburgh Supercomputing Center like working on this, and we have only been able to sort of look out to a few hundred neurons, not the 50,000 that Chung models. I also think another test of this is going to be how well this generalizes to other tasks and frameworks. So this is a really simple stimulus, a very simple task. Kind of for the purposes of this task, all the neurons do the same thing. And so we've been starting to do this with more rich tasks, more rich stimuli, and it holds up pretty well again, but we are just in the earliest stages of figuring that out. And then the last thing that we've done, we have just started collaborating with Caroline Runyon, who records using 2-photon imaging in mice, where she can identify a lot of cell types. And so there we can see what happens if you leave out different cell types and things. And that's just starting. There are definitely topological differences between cell types. And so I hope I'll have a better answer for that in like coming years.
Thanks.
It's a question from Puya.
Yes, what I see, Lucas's hand is raised, so I would rather have him ask his question.
Oh, you can go ahead. I can wait.
Okay, all right. Thanks for the great talk. I was gonna ask a follow-up question on the controllability measure, just making sure that I understand what the plots were. So from your explanation, I think it's the plot that you showed as the energy landscape is kind of like a probability distribution over the different neurons and activity between them.
Yes, that's right. That's mostly right. Yes.
Okay, then in that case, wouldn't it be a function of the data that you already have? Because we are always limited by the data that we're collecting, then how confident can we be at like that description being a correct description of the distribution of responses overall in general?
Yeah. So just to be really clear, this is a probability distribution over network states, meaning a vector of population responses, so not over number of neurons and that kind of thing. But you're of course right that this is only as good as the data we collect. Again, I would say that we're just in the early stages of chasing this sort of thing down. I think that these data sets, so you didn't see me talking about like the details of little bumps and wiggles, right? And that's very much on purpose. I would say that these data sets are not going to be especially great for answering your question because we just have very limited stimuli, that sort of thing. We are collecting a lot more data, recording from a lot more neurons, different brain areas, and many, many different naturalistic stimuli. And I think that that's a really important question. So put another way, I think we've only sampled a very small subset of the states that this network can be in, and we're trying to expand that a lot.
Got it, got it. Just one more follow-up question, if I may. So it sounds like this measure of controllability might actually be related to these attractor points in dynamical systems. Do you have anything to say about that? To me, it sounds like when the controllability is high, it's actually in a very sharp attractor point that doesn't allow it to go around and explore. But when the controllability is low, it seems like it's kind of flat space that the neuronal population can move around. Am I getting this right?
I think so. My intuition is exactly the same as yours. And to my knowledge, there hasn't been a lot of work looking at these things. Again, this data set is not especially great for looking at dynamics, but another thing that we haven't used is sort of in time, right? I was talking about cross-trial correlations, but as you said, the whole population has dynamics, and that will also provide constraints on the ability to move between one state and another. I think that's incredibly interesting, and yeah, that's a direction we'd really like to go.
All right, thank you so much.
Thanks.
So I guess there was a question from Lucas, right? So please put the questions in the chat because I'm not monitoring the hand raises, so if possible.
Yeah, sorry about that. I'll use the chat next time. Well, thank you for your talk. I mean, I'm very new to neuroscience, and I think I learned quite a bit. One of the questions I have is about those eigenmodes from Chung's model.
Mm-hmm.
Is that like, are those, is it like PCA where you're doing like it's a linear combination of the number of spike counts for specific neurons, and then you're just finding which linear combinations are like you know, most important, or is it some other, like
it is for all intents and purposes that. So you can imagine plotting the response of the population in a high dimensional space where every dimension is one neuron. So if we recorded 100 neurons, it's a 100-dimensional space.
Okay.
And the only difference between, so you could just do PCA on that. The only difference between what Chung did and PCA is that factor analysis, which is what she used, partitions that variability into variability that's private to one neuron versus shared. And so what this is showing you is the proportion of the shared variance that is accounted for in each dimension. But yes, the intuitions are the same.
Okay, awesome. And then if I have maybe another quick question about, you were talking about drawing the dots where each dot represents a neuron, and the distance between the dots was, you know, kind of opposite conceptually to the noise correlation.
Mm-hmm.
Are you, when you're drawing these pictures, are you trying to find like the minimum number of dimensions that you can draw that picture that where all the constraints are satisfied, or do you just find an example that works? Like, you know, I can draw, if I have 6 neurons and all these constraints, like 6 factorial constraints, then, you know, oh, I can draw this picture in like 7 dimensions. That's fine. Or do you try to find the smallest dimensional space that you can draw that picture with all the constraints satisfied?
Yeah, I love that question. You know, so Tevin, who does this work, he doesn't draw pictures. I draw pictures because it helps me to get intuition. And what the supercomputer is doing is not worrying about the dimensionality of that, is sort of taking whatever dimensionality it needs and just analyzing the relationships. And you can imagine that when you're looking for holes, putting it into the minimum number of dimensions is really not that important, because I could have a hole that's topologically equivalent to a circle and it exists in this flat plane, but I could also sort of bend it into a 3rd dimension and it's still topologically equivalent to a circle. And so for these particular measures, that doesn't matter. I think this is sort of an interesting space to explore using other methods as well. And I think questions about, for example, how high dimensional is that space compared to the maximum? How does attention change that dimensionality? I think that's pretty interesting, but we haven't really looked at that.
Okay, thank you.
Question from Shale.
Hey there. Thanks for the talk. It was great. And as someone with pretty much the same background as Devin, I was very, very happy to see this on the docket. I guess what I was curious about is that, you showed the slide where the kind of dimensionality or linear dimensionality kind of varying with past performance. And there's also a relationship with that and the topological measures like number of holes, which would probably also be like a direct dimensionality of computation. So I guess I was just curious, in terms of biological mechanisms, is there a way to actually check to see if there are physical circuits that are being created, or if it's just kind of functionally defined topological little patterns?
Yeah. I mean, the short answer is I would love to be able to do that. I know a couple of things, but not that many. So one is that you're absolutely right. It is an observation in neural data that even if you consider responses to a whole range of stimuli, for example, you record responses to the natural world, the activity lives in a much lower dimensional subspace than it would have to. And that's been shown in many species and systems. And I think that provides interesting constraints on neural coding. And then another question is, how do the circuit mechanisms what does that tell you about the underlying mechanisms? And are there relationships between sort of my holes and some sort of real circuit? What we do know from really good work by Tom Mersick-Flugel in mice is that there is a relationship between these noise correlation measures and actual connectivity. So he goes through and measures the synaptic strength that connects two neurons. That's the biology, the anatomy. And then he measures the noise correlation. And there is a relationship between those. Of course, those things, though, are more or less hardwired, meaning that if they're going to change, it's a much, much slower time scale than like shifting your attention around. And so when we see changes in the noise correlations, those are not about the anatomy, I think. So I think these are related to, but not identical to the anatomy. I would love to know what it means about circuit mechanisms. We also know that in cortical circuits, there are these motifs of connectivity that that are repeated, and that we think we know some things about what exactly is changing when you change something like attention. And so understanding the relationship between those is something that my collaborators, Chung Huang and Caroline Runyon, are pretty actively pursuing.
Great, thanks.
Okay, so we have hit the time limit, actually. I also had a lot of questions, but I guess I'll catch up with Marlene later. So thank you everybody for coming. And then once again, thanks a lot, Marlene, for coming and giving this fantastic talk. Thank you very much.
Thank you so much.
Thank you.